In blue is BARD1, BRCA1's partner protein. BRCA1 must first bind to BARD1 to form the dimer as pictured here from Clark and others. The dimer can then perform tumor suppression by a process called ubiquitination. Ubiquitin is shown here in square A from Pickard and Edens. Ubiquitin is itself a protein that modifies other proteins post-translationally to regulate biochemical pathways. Ubiquitination occurs when enzymes catalyze the creation of a covalent isopeptide bond between ubiquitin's terminal glycine and an amino group from the substrate. Usually, a lysine side chain contributes this amino group. How does ubiquitination work? In general, it happens in 3 steps, as diagrammed here from Pickard and Edens. 3 enzymes do the job. They're known as E1, E2, and E3. E1 activates ubiquitin. E2 conjugates ubiquitin, or UB. E3s, this is where BRCA1 protein comes in, as BRCA1 is an E3 enzyme. E3s are ligases, which brings the E2-UB complex together with the substrate being modified. According to Clark and others, ring E3 ubiquitin ligases, like BRCA1, act, quote, solely as scaffolds. Their job is to bring activated UB close to the molecule it's supposed to modify. In ring E3s, the specific structure responsible for ligase activity is the ring finger domain. The exons encoding this constitute a major site at which genetic tests check for mutations. Since cancer-predisposing mutations to this domain typically affect either BRCA1 BARD1 binding or the associated E2 enzyme, Clark and others say researchers conclude that ubiquitination is a key tumor-suppressing function of BRCA1. Substrates which BRCA1 ubiquitinates include estrogen receptor alpha, progesterone receptor, and histone protein H2A. Modifications impact gene activation, DNA repair, and DNA condensation, likely pathways for tumor suppression. It follows that mutations to exons 2 through 7 of BRCA1 gene may be clinically important in the development of breast cancer. According to Clark and others, the ring region's zinc coordination sites are sensitive to cancer-causing mutations. For example, replacing one of the coordinating histidine residues will affect folding and ubiquitination activity, and also predisposes to breast cancer. However, individualized information about cancer risk does come at a cost. When BRCA1 was first discovered, 3 main tests were available for individuals to determine their risk of developing breast or ovarian cancer. Single-site BRCA analysis cost $295, 3-site BRCA analysis cost $450, and comprehensive BRCA analysis with full gene sequencing cost $2,600. Despite initially high prices for tests, availability has increased since tests went to market, and cost-effectiveness is improving. Norum and others find that BRCA testing is effective and superior to family history alone, even considering the cost. Lim in 2018 implied that BRCA mutation testing is superior to routine clinical surveillance. Further research is warranted. Innovation will multiply the comprehensiveness and usefulness of tests, and competition will reduce prices over time. Ethicists and economists will need to think ahead about cost and information questions as total genome sequencing becomes an option for an increasing quantity of patients.